Hello everyone, welcome to today's webinar. I'm Catherine Dold with CNEN and I'll be moderating today's event. This webinar is titled Unveiling Solubility, Dissolution, and Permeability in Vitro, How GSK Sees the UV Light, and is being sponsored by Pion Incorporated. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with their mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look for the Help widget at the bottom of the screen or type your query into the Q&A box. If you get disconnected during the webcast, please log back in according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. Our speaker will answer them at the end of the presentation, and as your moderator, I'll be posing as many as time permits. Any questions that we don't get to today will be answered by email after the event. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinar, and that the webinar will be archived at CNEN online after the live webcast. Our presentation today is being sponsored by Pion. Pion is the leading provider of tools and analytical instruments that bridge the gap between in vitro experiments and in vivo outcomes. The instruments help companies confidently decide which drug substances and drug products to progress through the development pipeline while reducing risk and long-term investment. Products investigate dissolution, solubility, permeation, ionization, and absorption properties of compounds and drug products. Our speakers today are Wayne Matthews, Principal Investigator R&D, Drug Substance Development with the GSK Materials Science Group in the UK, and Carl Box, Chief Scientific Officer Europe with Pion. Wayne Matthews has a Bachelor of Science in Analytical Chemistry and is a pharmaceutical scientist with over 25 years of experience in the pharmaceutical industry, working for SmithKline Beecham and GlaxoSmithKline. He has extensive knowledge of developing new chemical entities and drug formulations for oral solid dose, injectable, and inhaled routes of delivery, and has worked across a broad range of roles covering early and late phase pharmaceutical development, analysis, drug delivery, bioenhancement, molecule selection, and high throughput automation approaches. He currently works as a principal investigator within the Materials Science Group at GSK. Carl Box was appointed as the Chief Scientific Officer Europe at Pion UK in 2020. He is involved in scientific and chemistry-related functions within the company, as well as supporting commercial activities and business development. His expertise is in the field of physicochemical measurements, where he has forged a successful career in the development of new instrumentation and assays for supporting drug discovery and development. I'll now hand the program over to our first speaker.
Today we will present an overview of the use of Pion in vitro dissolution testing methods within GSK for optimizing molecule selection in early drug development stages. I will start off with an introduction to Pion and the Pion product range. I will highlight instruments used by Wayne Matthews, our industry speaker, and his colleagues at GSK. I will then provide more information on the Rainbow Dynamic Dissolution Monitor, which is used for drug quantitation in many of our platforms, and also on the Pion surface dissolution imaging equipment. I will then hand over to Wayne Matthews, who will provide an introduction to GSK, the role of biopharmaceutics and in vitro dissolution, and the use of Pion methods for optimizing molecule selection in early drug development stages. Wayne will illustrate his talk with selected case studies before we finish off with a live question and answer session. Pion is a scientific instrument and services company that provides tools for understanding physicochemical properties, dissolution, and drug absorption at various stages of the drug development process. The headquarters is based in the United States, in Billerica, Massachusetts, about 1 hour north of Boston. Here we manufacture and assemble all of the instrumentation, as I will show you on the next slide. We also have another office in the UK, about 1 hour south of London. The UK is the centre of excellence for providing analytical services using all of the equipment manufactured by Pion. This slide shows the range of current products that Pion is offering. As mentioned previously, Pion provides tools for understanding physicochemical properties, dissolution, and drug absorption at various stages of the drug development process. This can range from PAMPA, pKa, log-P and solubility measurements during drug discovery, miniaturized low-volume dissolution and flux apparatus during pre-formulation and early drug development, through to larger volume dissolution and absorption tools during the formulation and drug product development stages. In addition, we supply novel tools for looking at dissolution at the surface of materials and for simulating the performance of subcutaneously injected formulations. The instruments that are circled are currently in use at the GSK site in Stevenage, UK, and these platforms will be illustrated by Wayne Matthews with selected case studies later in this presentation. Before handing over to Wayne, I want to mention the Rainbow Dynamic Dissolution Monitor. This is our flagship instrument for performing direct in situ concentration measurements in real time. It is a dedicated UV spectrometer fiber optic platform that can be integrated into a range of Pion and other manufacturers' products. It houses a deuterium lamp that connects up to 8 fiber optic UV probes that have an open window and a quartz mirror at the end of each probe. When the probes are inserted in a measurement vessel containing a dissolution medium, the light reflected from the quartz mirror is captured by a diode array spectrophotometer and used to determine dissolved drug concentrations. Each probe has its own dedicated spectrophotometer, and wavelengths can be simultaneously captured from 200 to 720 nanometers. Overall, the system is very robust and low maintenance, but its major advantages come with its capability of capturing lots of information about dynamic dissolution and dissolution absorption processes directly in situ without the challenges associated with offline sampling and analytical method development. We routinely capture UV spectra every 30 seconds and could record data every 2 to 3 seconds if needed. The Pion SDI2 is a flow-through dissolution apparatus that allows a user to understand the dissolution process close up to the surface of dissolving materials. There are 2 types of flow cells, a low-volume flow cell for pre-formulation groups and a large-volume flow cell for formulation and product development groups. This is modelled on the United States Pharmacopoeia Type 4 dissolution apparatus flow cell. The SDI flow cell incorporates quartz mirrors through which UV invisible light at selected wavelengths can be transmitted to a large area array camera detector with over 4 million pixels. Samples are inserted into the flow cells and dissolution medium flows over the samples, and the amount of transmitted light can be used to look at the dissolution process and the surface of the materials. In this way, we can obtain a detailed spatial view of the dissolution process at the solid solution interface, unseen by traditional dissolution methods. Now I will hand over to Wayne, who will describe the use of Pion tools within GSK and provide some example case studies.
Hello everyone, my name is Wayne Matthews. I'm a Principal Investigator within the R&D Material Science Group at GSK. I'm going to provide you with some insights into how GSK utilises UV-based dissolution tools to characterise and make decisions on the development of our molecules. I will provide a very brief introduction to GSK and how we typically apply a range of in vitro tools to characterise our active pharmaceutical ingredients. Then I will cover in more detail the techniques we have available to us internally to perform this type of work, an example of how it can be applied. I will then talk through 2 case studies in more detail. and finally summarise before hopefully being able to answer some of your questions at the end. For those of you who are not familiar with GSK, we are a global biopharma company with a range of vaccines, small molecule, and biopharmaceutical-based assets in our portfolio, covering a range of therapeutic areas, including HIV and oncology. We have 3 main R&D sites for small molecule and biopharmaceutical development, which are Stevenage and Ware, near to London in the UK, and Upper Providence, near Philadelphia in the US. Our R&D scientists use Pion equipment on each of these 3 sites to investigate and characterize the properties and behavior of new chemical entities. Images on the map indicate the Pion equipment we have available at each of the sites, and I'll cover the detail of these in subsequent slides. My current role is within the Materials Science Group at GSK, and I work closely with the other teams, most notably biopharmaceutics and drug product development, to assess new drugs for issues surrounding their absorption and hence their success as a potential medicine. GSK uses an integrated in vivo, in vitro, in silico approach to help understand the absorption of our molecules. This essentially means we take information from experimental testing in the lab, such as dissolution and solubility, and combine it with in silico pharmacokinetic models to predict different scenarios. This is then correlated with the data obtained from either preclinical species or in humans. Our aim within product development is to design medicines with the best possible performance for the patient that the medicine is intended for, and understand how the properties of the drug or the formulations we develop can potentially impact this performance. This slide shows GSK's typical approach to performing in vitro dissolution testing. These are the tests that are used to predict in vivo behavior in preclinical species and in humans. Our approach is based on being able to predict potential risks and issues as early as possible in development, so that problems later are avoided. As we move from left to right, the testing applied potentially becomes more complex, becomes less flexible, and significantly increases in both cost and the amount of material required. However, the trade-off here is that the test is possibly more biorelevant in nature. Pion UV dissolution tools, such as the Rainbow Profiler and Surface Dissolution Imaging System, can be utilized to rapidly screen properties of APIs with very small amounts of material before moving on to further assess the performance of the dosage forms at a larger scale, typically in more standardized USB 2 dissolution systems. This is done prior to testing in more complex in vitro systems such as the TNO TIM1 or Tiny Tim platform. Our goal is to be able to assess and predict the behavior of our molecules in as fast, efficient, and material-sparing manner as possible in order to make critical decisions at the right stage to support development of the product. Here we can see the range of techniques we have available across our GSK R&D sites. We have an array of what we would term exploratory dissolution equipment, i.e. those are not intended for compendial tests for QC, batch release, or stability indicating purposes. However, they may be useful in understanding issues relating to failure of these types of tests. These techniques are used to help characterize our drug substances and potentially any resulting drug products depending on the phase of the asset. To the left-hand side, the Rainbow UV Profiler, as previously mentioned by Carl, forms a significant part of the toolset and can be utilized in a number of different setups depending on the properties we are looking to investigate, such as solubility, dissolution rate, or permeability, and a combination of all of these properties. To the right, you have the Surface Dissolution Imaging equipment, which provides visualization of the solid-liquid interface as the drug substance or drug product dissolves or disintegrates, and can provide mechanistic information on these processes. In the center of the slide, we have non-Pion techniques that can also provide additional information to help us characterize the behavior of the API during dissolution. To provide a better mimic of the human gastrointestinal tract and its influence on the dissolution of active pharmaceutical ingredients, solubility and dissolution experiments can be conducted in media that simulates the observed properties of in vivo fluids. These are comprised of various bile salts and phospholipids to simulate fasted and fed state for both the gastric and intestinal environment. In regards to the use of these fluids for UV-based dissolution systems, some components that reduce the light transmission or heavily absorb UV light, such as lipids and proteins, can prove problematic, especially for UV imaging. The use of a media blank correction channel on the Rainbow Profiler system can sometimes improve this situation. However, if the media is completely non-UV transparent and the amount of undissolved particles is too high within the vessels, then dissolution performance can potentially be measured indirectly, such as via microflux permeability analysis, thereby correlating the amount absorbed with the dissolution rate or solubility obtained. The microdisk, or rainbow profiler, is Pion's commercially available fibre optic UV dissolution system. This system is designed to monitor the concentration of dissolved materials in a range of liquids without the need for removal of the samples to test offline. This allows for the use of very small amounts of material and testing volumes. The utility of these small-scale dissolution systems have been demonstrated in several studies for a variety of applications, including the measurement of intrinsic dissolution rate, dissolution permeability studies, and the screening of formulation performance. Microdisc fibre optic probes can be placed into any vessel containing a liquid, and the UV path length is adjusted based on the expected concentration or the UV absorbance of the sample. Additionally, rapid sampling times mean the system is typically used for analysis of API to study aspects such as differences in molecule version or form, particle size, formulation impact, and precipitation risks, which can be missed by systems with long sampling intervals. Second derivative spectroscopy is typically used to build up a concentration profile over time. The system can be used in volumes ranging from 1 to 20 ml and assess powder and suspension samples. Here we can see an example of the dissolution and precipitation processes that are captured by the fibre optic UV-based dissolution systems. The API here was assessed as a number of alternative salt forms to help improve the low solubility in a gastric environment. However, solubility analysis showed all of the salts to be exactly the same as the free form of the compound. The microdisc fibre optic UV system captured the differences in dissolution of the various salts and their subsequent supersaturation and precipitation events that were missed by simple solubility analysis. These events can also be seen qualitatively using a digital imaging platform and demonstrate the link between the behaviour occurring within the dissolution media and the quantitative measurements made. The rainbow profiler can also be used for the measurement of intrinsic dissolution rate, or IDR. Historically, the use of the Wood's rotating disk apparatus was used to collect IDR data on drug molecules using large amounts of powder and dissolution media volumes. IDR is an intrinsic property of the molecule when tested at a particular set of experimental conditions such as pH, compression force, and agitation rate. IDR uses API compressed into disk to maintain the constant surface area during the experiment. This can be an excellent method for comparing the performance of different molecules, different forms of the same molecule such as salts or polymorphs, or for investigating different physical properties such as particle size. The Pion Micro IDR setup allows this test to be performed on 5 to 15 milligrams of material, a significant saving over the traditional Woods apparatus. This generated IDR of very poorly soluble compounds can be extremely challenging due to the limits of detection in solution and issues with compression. And so the Pion AE Pro software used for dissolution analysis can also be used to estimate this value for a powder sample via a set of algorithms. Here's an example of a GSK molecule assessed for intrinsic dissolution rate. In this instance, the molecule was found to exist as multiple polymorphs, one of which only presented itself upon compression of the form to make tablets. The second form identified did not undergo any polymorphic change or compression, but all the previous in vivo studies had been conducted with the first form. The question was asked if there was potentially a risk around the change in performance by switching from one form to another. Initially, due to issues around compression of one of the forms, to maintain a pure form, the powder IDR method was used to compare the samples. You can see this in the first graph. Both forms 1 and 2 were observed to have comparable dissolution rate, as shown by the profiles and corresponding IDR values. The two forms were then also tested in the more standard compressed disk approach, with the compression force previously assessed not to induce any formation of group B. Here you can see a slightly lower dissolution rate for form 2, but this is not regarded as significantly different, and therefore the risk of switching between the forms is low. Additionally, there is excellent agreement between the powder and disk methods used. The last method that can be used with the Pion Rainbow Profiler is a simultaneous measurement of dissolution permeability, also known as absorptive flux. This measurement predicts the potential bioaccessibility of the molecule or formulation being tested by measuring the amount passing through an artificial membrane based on a pamper setup, post-dissolution of the material in a selected media. 2 cells are used for one sample in this analysis, with one fibre optic probe in each. The first cell is the dissolution chamber. The second connecting chamber is separated by a lipid-covered membrane representing the gastrointestinal tract. Higher amounts determined in the absorption or acceptor cell suggest an increased amount of the drug available for absorption. This can be used to investigate and rank order different APIs or formulation types. In this GSK example, of the use of the Microflux Permeability test. A compound with high solubility in both gastric and simulated intestinal fluid was assessed for dissolution and permeability in either a fasted or fed state to determine possible differences when dosed to patients. The dissolution results in the first cell were as expected, with the molecule rapidly dissolving. However, the resulting flux permeability was observed to be significantly lower in the fed media compared to the fasted, therefore displaying a more unusual negative food effect. This was the opposite of what was expected based on the solubility and dissolution analysis of the compound. This difference was also seen in human studies, both fed and fasted. It was only able to be predicted by using a dissolution permeation setup, such as the Microflux. Surface Dissolution Imaging, or SDI for short, allows visualization of the dissolution process of the API as it interacts with the media. This is achieved via a 4.2 megapixel camera attached to a flow cell recording UV images as dissolution of a solid material occurs under a range of different conditions. When run at a certain UV wavelength, false color images denote the level of UV absorbance obtained and can be used to determine the concentration in solution at every point in this image. The latest version of the instrument, the SDI2, uses UV and visible light sources and has both a compact flow cell for assessing API dissolution and a larger flow cell for visualization of dosage form dissolution. Both of these can help elucidate the mechanisms occurring during the dissolution process. In this example of surface dissolution imaging, an early screen was performed to determine whether salt formation would improve dissolution of a polysoluble compound. Standard solubility testing showed all salts to be the same as the parent free form of the molecule. However, the UV imaging dissolution test provided additional dissolution rate information on the different versions. This clearly showed the sulfate salt of the molecule to exhibit superior IDR to the parent and to the other salts obtained. No obvious disproportionation was observed for any of the salts in the IDR test, as indicated by no clear change in the dissolution rate. This was also confirmed by solid-state analysis of the API disks post-dissolution testing. Sometimes, the images themselves obtained by UV dissolution imaging are sufficient enough to assess the behavior and performance of a molecule. Here, the free base of a compound was observed to swell and gel significantly in acidic media, whereas a hydrobromide salt did not. Additionally, the salt showed a superior dissolution rate over the free base and was therefore selected for progression. In this particular test, although the mesylate salt of the molecule appeared to have an even higher dissolution rate, this was then observed to disproportionate back to the free base before the end of the test and resulted in the same gelling behavior. This gelling behavior can be clearly observed in the UV images obtained from the different versions and enabled an improved understanding of the dissolution failure for both the free base and the mesylate salt when run as larger scale dosage forms. So, we've seen there are a range of techniques available for characterizing the solubilization, dissolution, and permeability behavior of a molecule. The ideal goal is to apply these techniques as early as possible in development to help identify potential risks to progression. Our aim in very early development, when small amounts of API are available, is to predict the behavior and performance of larger scale batches of drug substance, i.e. moving from a microscale to a macroscale, and then to ultimately predict the performance of an API or formulation in an in vivo setting. A good example of this early prediction of performance at small scale is shown here. The compound shown earlier, as part of the surface dissolution imaging technique, was tested as both the free base and the hydrobromide salt, using the UV microdissolution system. This was then eventually run in a significantly more complex TNO-TIM1 gastrointestinal model. In both cases, the small-scale microdisc was able to predict the results of the TIM1 analysis based upon the gastric pH sensitivity of the free base and the resulting improved performance of the hydrobromide salt form. A recent addition to the in vitro toolbox is the Pion Mini DT, or Mini Dissolution Tester, based around a miniaturized version of the conventional USP2 dissolution platform. In this example, the standard mechanistic stirring of the microdisc vessel in a 20 ml vial was able to replicate the solubility of the compound, as seen via a shake class solubility analysis. However, running the compound at a small scale on the Mini DT using a volume of 100 ml and a paddle speed of 100 RPM was able to pick up a significant particle size sensitivity. This was also clear to see from both biorelevant dissolution analysis with standard USP2 and when run on a TNO-TIM1 system, suggesting that particle size reduction may be required for the compound. We now come on to the first larger case study, which looks in more detail at selection of an appropriate version of a molecule for development. This particular compound showed very low solubility as a free base, and therefore a salt screen was conducted in an attempt to improve the solubility. The sulfate salt of the molecule gave the highest increase in solubility, closely followed by a crystalline tosylate salt. The corresponding UV microdissolution of the two salts and the free base showed the sulfate salt to be clearly significantly superior, and therefore this salt was scaled up to perform further characterization and analysis on. Upon scale-up of the sulfate salt, USP2 biorelevant dissolution testing was performed to confirm the choice of the version of the further progression. The graph here indicates the dissolution analysis of the sulfate salt and the free base from as 30 mg of API in HPMC capsules. Data obtained from this method ran as both a two-stage dissolution from gastric to intestinal fluid and as a single-stage fast-state intestinal fluid alone shows the dissolution of the free base to be consistently higher than that of the sulfate salt, the opposite of all the previous data obtained. Further analysis of the solid residues post-testing confirmed the material was concordant with the input, and that the sulfate salt had not undergone dissociation back to the free base. Neither had the analyst performing the test simply mixed the samples up. Since no consistent set of data could be obtained for the different versions using multiple tests, and to manage an expected higher dose range in initial safety testing, an additional bioenhanced option of an amorphous spray dry dispersion was developed, whereby the API was stabilized with HMCAS. This was again run on the UV microdissolution system and compared against the previously identified crystalline salts and the free base of the molecule. In both a single and two-phase small-scale dissolution test, it is clear that the spray dry dispersion has a significant performance advantage over the salts. It was also noted that the tosylate salt exhibited a significant particle size sensitivity, since the dissolution rate was very slow in comparison to that of the sulfate, despite having similar solubility. Size reduction of the salt via micronization improved performance. This was then observed to be similar to that of the sulfate. To determine whether an STD was required and whether either of the salts were actually superior to the free base, they were tested in both the TNO-TIM1 gastrointestinal model and in preclinical animal species. The microdisk correctly rank ordered the different samples when compared to the TIM1 and animal data, indicating the sulfate salt was indeed able to provide a high level of absorption in comparison to the free base, whereas the USP2 method failed to predict this. However, despite showing the correct rank order for different samples, the microdissolution test appears to be overestimating the performance of the STD in relation to the salts, especially the sulfate salt, which looks almost comparable to the STD in both rat and dog studies. Further UV dissolution analysis, this time with the addition of absorptive flux, to provide an estimation of the permeability, was performed on the different versions and the spray dry dispersion in biorelevant bed-state media. Although the initial dissolution results obtained show a similar trend to the one observed in the previous small-scale testing, the additional measurement of flux permeability, as opposed to just standard dissolution testing, provides a much closer correlation to the results obtained in the TIM1 and in vivo animal performance. This suggests that a spray dry dispersion may not provide a significant advantage over the sulfate salt, and that the salt alone will provide adequate absorption. Based on this information, and other manufacturability considerations around the scale-up of the STD, the sulfate salt was then chosen for progression. As a follow-up test, the sulfate salt was then assessed for potential food effect, using the same dissolution permeability approach as used to compare the STD against the salts. As opposed to the previous GSK example, both the dissolution performance and the corresponding flux permeation values predict a high likelihood of a positive food effect for this compound. Additional testing in the TNO-TIM1 system also showed the same trend. This again demonstrates the predictive power of the smaller scale and less complex UV dissolution analysis. And so, selection of the sulfate salt was then confirmed. Intrinsic dissolution rate analysis was then performed on the different versions for completeness. You can see again the powder dissolution profiles run on the UV microdissolution system that shows a large difference between the sulfate and the tosylate salts. However, when the IDR test was run, on compressed disks of each version, we showed the toslate to have a slightly higher dissolution rate in comparison to the sulfate salt. Did we do something wrong? Is the IDR test incorrect? Should the sulfate salt not have a higher IDR in comparison to the non-size reduced toslate salt? During further tablet development trials, the different versions were then compressed as either micronized powders or as a blend containing excipients. These compressed samples were then broken back up to a powder and compared against a non-compressed sample in a UV microdissolution test. The results obtained show the sulfate salt has a much slower dissolution rate post-compression in comparison to the tosolate salt. It was subsequently found that under compression the sulfate salt was losing crystallinity and forming amorphous glass. It was extremely non-porous, thereby slowing down the dissolution process. Powder X-ray diffraction patterns for the sulfate clearly show the loss of crystallinity. This same behavior was also observed on a larger scale tableting system, showed the sulfate salt was susceptible to losing its crystalline structure on compression, and amorphous film was found to form on the surface of the tablet. Based on all of this data, the toslate salt was then selected for further development. To summarize the results from case study 1, fiber optic dissolution on powder and suspension samples correctly predicted the rank order of different versions of an API, which was not possible using conventional USB tube dissolution testing. Small-scale dissolution analysis also correctly predicted the performance of an amorphous solid dispersion. However, only the use of a dissolution permeation model, such as the Microflux, was able to obtain a good estimate of the relative differences between the spray-dry dispersion and the crystalline salts. Lastly, the use of a small-scale intrinsic dissolution rate analysis highlighted an extra risk of amorphous generation of one of the salts, which enabled selection of the most appropriate salt from both a bioaccessibility and also a manufacturability perspective. Case study 2 shows the analysis performed on a compound exhibiting highly pH-dependent solubility behavior. The change in solubility is typical of a weakly basic drug, where solubility can be extremely low at higher pH values. This particular molecule shows a significant drop in solubility moving from what would be considered a typical healthy volunteer gastric pH of around 2 to an elevated gastric pH of approximately 4 or higher. It would therefore be predicted that in volunteers or patients with a high gastric pH, there could be the potential for reduced or variable bioavailability. When dosed in humans, this API was found to exhibit hugely variable and much higher than expected absorption. In some cases, the absorption seen was high enough to trigger a stop to the study based on safety concerns. The question was asked as to whether there was anything that could be done to reduce the level of availability observed in vivo and provide a potential option to restart the study as quickly as possible. 3 different options were evaluated to determine possible alternative approaches to reduce the variability in exposure and reduce the very high absorption seen. The first was to assess particle size reduction and increased dissolution rate to ensure rapid solubilization in the stomach. The second of these was to remove any potential differences in the gastric pH of the subjects by dosing the drug with an acidic drink, thereby normalizing dissolution response. The next set of options was to determine whether the rate could be slowed down to prevent higher levels of absorption, either by dosing with food to slow gastric emptying, or by utilizing enteric capsules to release the drug in the intestine, where it would be expected to have a low level of solubility, and hence the permeability would be reduced. The final of these 3 options was to determine whether an alternative form of the molecule, such as one with a lower level of solubility to reduce the high absorption, or a salt to potentially overcome the expected gastric pH variability, could be used instead. For the first option of particle size control, the API was assessed using intrinsic dissolution rate analysis. The IDR plots here on compressed disk clearly show the extreme pH dependency on solubility. Dissolution rates above a pH of 2 are reduced approximately 30-fold. This would clearly have an impact in vivo if the subject's stomach pH was elevated, or potentially provide much higher absorption if their stomach pH was low. It is also clear that reducing the particle size via micronization, as seen in the second graph, does not appear to have a significant impact on the dissolution rate of this API, as observed by the very similar dissolution profiles and the corresponding IDR values obtained. Additionally, further solid-state analysis of the compressed disk from the micronized API post-dissolution at pHs 3 and 4 suggested the chemical form of the API was slightly different to that of the input. Repeating the dissolution analysis of the particle size reduced sample versus the initial first-time in human input API as powders at different gastric pHs again shows a large change in dissolution performance, as would be expected for this molecule. However, it still does not suggest a significant improvement to dissolution rate with a reduction in particle size. In this experiment, it was also noticed that the micronized material appeared to be reducing solubility over time in both the pH 3 and pH 4 conditions, potentially adding to the Raman solid-state data previously obtained on the compressed IDR disk, suggesting that a change in chemical form was occurring during the dissolution process. A further assessment of the batch of API used for the human study in comparison to previous batches prepared suggested that the drop in solubility could be due to a change in form of the material. The API was anhydrous, and therefore it was postulated that a hydrate of the molecule was being formed with a significantly lower solubility. Hence, this is what was precipitating over time in the experiments. The level of supersaturation present in the system and the amount of undissolved API present was altering the kinetics of this conversion. The change to the hydrate was picked up by the UV analysis due to an increased level of stability and greater scattering of light and the needle-like particles present, in addition to an overall reduction in the solubility. When comparing the different API batches, it's clear that the use of micronized material increases the kinetics of the turnover to the less soluble hydrated form, most likely due to a very slight increase in dissolution rate, meaning supersaturation is reached faster, and a greater surface area, resulting in a larger number of particles available to seed the system. Dissolution data also picked up the first-time in human batch is more susceptible to hydrate formation, possibly as a result of an increased purity. Isolation of the hydrated form of the molecule clearly showed it has lower solubility. This could potentially occur in vivo and could therefore be a cause of the variability. Subsequent analysis of the micronized and non-micronized API in the tiny tim gastrointestinal model show a very similar trend to small-scale UV dissolution experiments, and the micronization does not appear to offer a significant improvement in bioaccessibility. Option 2. Dosing the API with an acidic drink to normalize stomach pH and obtain a similar dissolution rate and solubility across different subjects was then assessed. For this experiment, we selected the most acidic and widely available drink possible, Coca-Cola. UV dissolution testing was then performed with the API at various gastric pHs and with the addition of different types of Coca-Cola. Profiles obtained clearly show the addition of Coke effectively normalizes the pH of the simulated gastric fluid, thereby increasing the solubility of the API to a level similar to that observed around pH 2. It is also thought that the Coca-Cola also has a solubilization effect on the API itself. The same effect was observed in a corresponding tiny tim analysis dosing the API with Coke in an elevated gastric pH scenario. However, the API only exhibits a small change in bioaccessibility when dosed with Coca-Cola, suggesting this effect may not be significant. One of the main issues in prediction of precipitation of weak bases is dose proportionality when larger doses are used in vivo. This is especially true when performing small-scale dissolution experiments without an absorptive sink, which can typically overestimate the precipitation potential. In this first experiment, using a dissolution permeability microflux setup, the API was dosed with and without Coca-Cola at elevated gastric pHs. Although the Coke is seen to effectively reduce the pH and increase the solubility of the API, this is then observed to show a rapid precipitation when moved into an intestinal pH media composition, such as passive, as would occur post-gastric emptying. The level of flux obtained suggests no significant improvement in the permeation would be observed in this scenario. In a similar way, the TIM-1 test also showed less of a difference than expected between dosing with and without Coca-Cola. There could therefore potentially still be a correlation between these two methods. One way to account for the different volumes available in small-scale testing compared to in vivo is to reduce the dose and concentration accordingly. In this experiment, the same dose was reduced by a factor of 5 to account for a 50 ml volume instead of the typical 250 ml, usually accepted as the initial volume of the stomach after ingestion of a glass of water. The experiment was performed using a two-stage dissolution setup, with the gastric phase being performed in the Mini DT dissolution system with 50 ml of SGF at 3 different pHs. After 30 minutes, a small aliquot of the suspension was then transferred to a flux cell containing passive media. The experiment shows an improved correlation with the gastric pH compared to the high dose concentrations previously tested, and the pH2 sample can now be observed to change its rate of flux when precipitation occurs. The pH4 sample also appears to have a similar flux, since the API has likely formed the hydrate in the gastric phase, which is why no subsequent precipitation is seen in the intestinal media. This suggests the initial gastric pH may not have a significant impact to influence the overall absorption of the API. The third option to help reduce and normalize the absorption in humans is the dose of food. Comparison between fasted and fed state media at small scale suggests only a very small change in the rate of precipitation. This is unlikely to be significant in vivo, although it is possible the addition of other components such as lipids and enzymes could further prolong the nucleation times in the presence of food. To fully represent the differences in gastric emptying times in a fed state, a transfer dissolution model would need to be used, using a syringe pump or similar to slowly add the gastric contents to the intestinal phase. Such a system is not available for the microdissolution platform here, but can be used on the Pion Inform system also available. Additional testing in the tiny TIM shows that whilst Tmax could be impacted in the presence of food, the difference to Cmax and AUC is likely to be minimal. Another way of reducing the level of absorption seen in vivo is to release the API in the region of the gastrointestinal tract where it has lower levels of solubility, and hence enteric capsules were assessed as a potential option here. The Mini DT was again used as a small-scale dissolution test, running a two-stage analysis from gastric fluid to fasted state intestinal fluid. The resulting profiles indicate that the lower solubility of the API in the intestine could reduce absorption. However, it is also likely to increase the level of variability, since disintegration and rupture times for enteric capsules will be more variable. Standard passive media uses a phosphate-based buffer, and enteric dosage forms are known to take longer to dissolve when run in biorelevant carbonate buffer systems that reflect in vivo. This variability is also observed in the TIM1 analysis, suggests this approach may solve one issue but enhance another. The last option available was to dose a different form of the API in an attempt to change its absorption profile. The hydrated form of the API was isolated and tested in the same two-stage dissolution permeability method as used previously. It's clear the absorption is significantly reduced by using the hydrated form. This could possibly lower the Cmax and additionally reduce the variability, assuming that hydrate formation occurs in vivo for the unhydrated form. The use of a salt was seen to significantly increase the dissolution in elevated pH gastric environment, thereby normalizing the impact of the stomach pH. But this appeared to have very little impact on the resulting level of flux, performing in an almost identical way to the free base anhydrate sample. In any case, neither of these options were preferred due to the significant delays that would result in preparing new materials to be dosed in the first time in human study. Conclusions from case study 2. Small volume UV dissolution analysis was used to evaluate API properties and behavior, which can be unseen by other methods. In this case, changes in particle size were shown not to be significant for a BCS2 weak base. Additionally, in situ fiber optics with rapid automated sampling can pick up changes in concentration due to precipitation and conversion to a different or less soluble form. The rainbow UV system works with Coca-Cola. Impact of gastric pH sensitivity can be assessed using simple dissolution experiments. Miniaturized systems can be difficult to assess the impact of gastric emptying times or precipitation potential, especially at high doses, but this can be normalized by testing proportional concentration based on the scale. UV dissolution experiments in the main provided the same information as was gathered by the TNO TIM1 platform. In the case of this compound, size reduction was not observed to improve absorption and may actually cause conversion to a hydrate, thereby significantly increasing the variability further. Dosing with an acidic drink, i.e. Coca-Cola, did normalize the gastric pH, resulting in a slight improvement in the bioaccessibility. Enteric capsules, although likely to reduce Cmax, would probably increase variability and were not selected. Repeating the first time in human with either a hydrated form or a salt would potentially reduce variability in vivo, but could also decrease or increase the Cmax respectively. and is not a quick option for this project. Dosing with food is predicted to delay the gastric emptying and could potentially slow down the absorption. However, there's no significant indication of this from the in vivo data generated. To summarize, GSK currently uses a range of UV-based dissolution methods to help measure and assess potential risks to development and help select and optimize molecules for progression. Small-scale methods shown here can be used flexibly to assess a wide range of samples, from drug substances to full solid oral dosage forms and everything in between. Rapid sampling via UV approaches, either as fiber optic or surface dissolution imaging, can provide enhanced understanding of the mechanisms involved in dissolution and further help to troubleshoot and understand issues. Combining permeability with dissolution can be key to determining differences between samples and formulations, especially when solubility in the dissolution phase is limited. Overall, GSK has seen very good correlation from these types of methods to more advanced in vitro systems and to in vivo data, further enhancing their application. I'd like to take some time to acknowledge the people both within GSK and also at Pion who have enabled me to make this presentation possible.
Thank you, Carl and Wayne, for a great presentation. We have a few minutes left for questions. A reminder to our audience, you can submit your questions in the Q&A box at the bottom of your screen, and any questions that we don't get to today will be answered by email after the event. Also, please note that Andrew Kennedy, an Analytical Services Group Leader with Pion, will also be answering questions this afternoon. So let's get to our first question here. Has GSK considered the use of UV-based dissolution techniques for GXP applications? Who'd like to take that one?
So I can take that one, coming from GSK. So the answer really to this is no, we haven't. And in the majority, this isn't because we don't value the use of the system in that way, but it's mainly because if we were to validate our factory methods with an in situ fiber optic UV type method, we would have to ensure that all of the factories making products around the world were equipped with the same type of system in order to transfer technical methods, and that's why we don't typically use it for GMP purposes.
Okay, thank you. Next up, for the two-step U-flux or microflux analysis, do you have a best practice for experimental setup? Do you worry about permeation during the FAST-GF first step causing a donor depletion before the second step, Feren's media switch, can begin?
So I can take that one as well. So we typically run that type of test in two different ways. The first way is to have the SGF inside the flux donor or dissolution cell, and in that case we actually have the volume of the SGF below the level to which it can it's in contact with the membrane, and therefore nothing can permeate through until we add the intestinal media into the cell. The second way is that we do it as the SGF part, so the gastric part, offline effectively. So we do that in either a separate vial on another microdissolution bath, or we, as an example showed in the presentation, we take it from the Mini DT, the miniature USB 2 system, and we use that as our gastric phase instead.
Okay, thank you. Next up, how can you add powder to the dissolution media to check powder dissolution without making a compressed disk?
Yes, so I can answer this one. So there are a couple of ways in which the powder can be added to the dissolution medium. The first one, the most simple way, is simply to dose the powder directly into the vessel from the top. They have an open top, and there is room around the probes with which to add the sample powder. The alternative is to actually dose your sample powder into the assay vial dry, and then when the assay begins is to dose your medium on top. Now this way round can help with wettability, particularly if the powder is expected to be hydrophobic. Helps to disperse the powder throughout the medium. The microdisk platform and the MB8 has a delayed data collection start and a delayed stirring start as well, so that can be automated to start with a 10 to 15 second delay to allow you time to dose either your media or your powder at the start of the assay.
Okay, thank you. Just a reminder to our audience, you can submit your questions in the Q&A box at the bottom of your screen. Next up, how is the suspension placed in microdisc? I did not see any sample holder in the picture.
So suspensions are typically added in directly to the vessel using a kind of a standard displacement pipette. So we would take an aliquot of suspension and dose that directly to the vessel already containing the media is a typical practice.
Okay. In addition to microdisc, can microflux also be used for suspension dissolution evaluation?
Yes, very definitely so. Yeah, so we also run this type of tests when performing assessments of suspensions for preclinical studies. So in animals such as rat or dog studies, that's something we do quite frequently. So yes, it also works for suspensions.
Okay, thank you.
So can I just add a little bit to that question as well? Using the suspensions in a microflux setup can provide an advantage as well over the straight microdisc, which Wayne actually alluded to during his slides. When we're adding suspensions, particularly to the small volume, you can end up with a very turbid dissolution medium, which can pose some challenges to the in situ UV. We measure with the microflux, you do get an idea of dissolution behavior, so indirectly as a result of the flux measurement, which remains clear throughout the assay. So even if you're fighting against turbidity from your suspension, the flux data should provide you with some indirect measurement of that dissolution behavior.
Okay, thank you. Next up, do you use any mathematical approaches when assessing UV measurements to assess dissolved drug concentration?
I'm not 100% sure on what that question is asking. Andrew, anything from you on that one?
Yeah, so ultimately the data from the, if we're referring to the microdisc here, the data that you obtain is concentration against time data. So a pretty standard dissolution curve, and you can perform any sort of dissolution curve analysis that you might obtain from traditionally sampled data that would also apply to these measurements here.
Okay, thank you. Next up, what suggestions do you have for improvements or enhancements for the different platforms?
Well, in terms of the rainbow profiler type system, I think potentially something that could provide an automated transfer from a secondary system, such as something like the Mini DT, into the microdisc, but I think that would potentially be overcomplicating things. I think for the SDI platform, the recent SDI2 is kind of what we wanted it to be based on the recent refinements. And I think the addition of the Raman analysis to get solid state information is something that we really need to start utilizing within the industry a lot more. Yeah, that's probably it.
Okay, thank you. Is the data from the SDI typically analyzed in a quantitative way, or is it for qualitative analysis?
Yes, I can take this one. So data from the SDI2 is absolutely quantitative. Not only do you get those images that we saw in the slides to provide those qualitative sort of mechanistic analysis, but so using sections of the image, we can analyze the absorbance that comes from the dissolved drug in order to calculate concentration, as well as using image analysis tools to assess the size of perhaps a tablet as it dissolves and how that changes. So we can get an understanding of like the rate of disintegration, for example, or the rate of swelling of a tablet in perhaps an extended release dosage form.
Okay, thank you. We have time for just one more question. Are there any molecules or applications you have tried that do not work with these approaches?
So the obvious one is that molecules without a UV chromophore, for obvious reasons, don't work. So that is the limitation, but that's probably the limitation for a lot of UV type techniques anyway. That's probably all that we've found in general that we really can't get any data from.
Okay. Any final thoughts? Nope. Okay. Well, that is all the time we have today. Thank you again to our speakers and our participants. And thank you to On24 for technology and production services. And thank you, Pion, for the sponsorship that made this interactive webcast possible. Be sure to check CNEN or CNEN Online for information on upcoming webinars. For CNEN webinars, I'm Catherine Dole. Goodbye.